I'm Greg Gilbert, and on behalf of my collaborators, Asa Conover and Ingrid Parker, I'm excited to talk to you a little bit today about work we've been doing with fungal metabarcoding to study networks of plant-fungal associations in complex plant communities. As plant ecologists, when we see a scene like this, we think about the diversity and the beauty of the plants in those habitats. But we also know that all of those plants are filled with fungi. And what we are interested in is trying to understand what's going on in those plant microbiomes. How many fungal species are living inside the plants? How does fungal diversity track plant diversity? Are those fungi specialized on individual plant species or on different habitats? And do different guilds of fungi behave differently? Now, when we think about fungi on plants, our minds often quickly go to the destructive impacts of fungi in causing disease. And those impacts are certainly important, but fungi inside plants also serve as mutualists, as commensals, and as parasites. And in fact, if we take what seems to be a very healthy looking plant and we take a tiny little piece of its leaf and surface sterilize it by putting it in bleach to get rid of any fungi that might be on the surface, then take those little leaf pieces and put them in a growth medium for fungi, in a few days, somewhere between 20 and 100% of those leaf pieces will show fungal growth, depending on the host plant species and the habitat in which it was growing. Now, we can use those cultural approaches to study the fungi associated with plants. And for fact, Ingrid Parker and I spent 4 years at Bodega Bay composing this table that shows 23 species of fungi that we found in the leaves of 17 co-occurring species of clovers using those cultural techniques. Some of those fungi are very common and widespread across lots of species, and some are more specialized. We identified those species with the help of using the standard DNA barcode for fungi, which is the ITS sequence, which is this internally transcribed spacer region between the small and large subunits of ribosomal DNA. And we use these barcodes because the diversity of fungi is so vast. There are several million fungal species out there. Most of them don't have names yet or sequences deposited for them, unfortunately. In fact, in the USDA database of plant fungus associations, there are already 99,000 fungal taxa listed. And even in a single crop species, we can find as many as 500 or more known fungal symbiont species. Now, those numbers are far in excess of what we see with larger organisms like mammals and birds and amphibians, and even thousands of plant species. There's only 17,000 plant species in all of North America. So the diversity that we're dealing with is quite astounding. And if we want to study fungal communities in complex plant communities, taking a cultural approach simply isn't feasible. So if we want to know what fungi are doing in a farm like this, or in a grassland community, or in a forest community, we've moved to a metabarcoding approach. And we're asking questions like, how do fungal communities differ across these habitats? Or how does fungal diversity relate to plant diversity? And how does fungal host range, the number of different hosts on which a fungus is able to grow, shape those plant fungal ecological networks that could form? Here at UC Santa Cruz, we are very fortunate to be at the north end of the Monterey Bay, right here on the central coast of California. And on the UCSC campus on the right, outlined in pink, we are doubly fortunate to have a tremendous living laboratory on the campus. On it, we have at the north forest habitat, mixed evergreen forest, including the forest ecology research plot. In the middle, several beautiful meadows, extensive meadows, and at the bottom, a certified organic teaching and research farm. We took advantage of this living laboratory to study the fungal communities associated with plants in these complex communities. To do that, we went into each of the habitats and we found all of the different species that were there that occurred at least 5 times. And we took 5 samples from individual plants that were separated by at least 5 meters to try to get a representation of what fungi were associated with leaves across the whole population. We surface sterilized those leaf pieces with ethanol and bleach so that we would only study those inside the leaves, extracted the total DNA and amplified the fungal ITS region, and then used PacBio sequencing to get the full 600 base pair ITS region for all of the fungi living inside the leaves of all of those different plant species. Now, standard DNA barcoding for fungi is great, but it does have some limitations. First is that you need to be able to compare those barcodes to a database. And there's been a long global effort to build those ITS sequence databases, but unfortunately, there's very poor quality control in most of them, with lots of misidentifications or simply no identifications in the fungal sequences. There is a great effort in the Unite database to provide sequences of validated fungal taxa for ITS sequences, and that's been tremendously helpful, but it only includes a very small proportion of the taxa that you are likely to find in these sequencing efforts. So we rely much more heavily on using OTUs, operational taxonomic units, taxa of fungi that we create using phylogenetically agglomerated taxa from the sequences. If we look at the species accumulation curves that we find on those samples, the x-axis is the cumulative number of host species in a particular habitat and the cumulative number of fungal taxa, OTUs, that were found as we add more and more host species. This is shown for the 3 different habitats separately: green for the forest, blue for the farm, and orange for the meadow. And you can see that there's a tremendous amount of diversity, which is what we usually find looking at fungal communities, and that the more plant samples that we took, the more fungi we find. Lots and lots of diversity out there. If we break that down into major guilds of fungi, we see pretty much the same pattern all the way through. The filamentous microfungi, these ascomycetes that are common on plants, mushrooms and their allies that live inside plant leaves, the biotrophic rusts and their allies, and smuts and some really interesting yeast allies. All of them show these similar patterns. And we'll come back and talk a little bit about some of these guilds later in the talk. One of the big questions we have is about the fungal host range. And so that is how many different plant species of fungus is able to colonize. And these histograms here for each of the 3 habitats show on the x-axis the number of different plant host species on a log 2 scale on which of a particular fungal taxon was found. And then the bars are the number of taxa in those host range bins. So the top bar, the big bar on the left, represents those fungal taxa that were found only on one host. And that's 39% of all the fungal taxa. The other 61%, of course, were found on many more species. Now, our sampling design does not allow us to distinguish between host specialized and rare fungi. So we won't be spending more time thinking about that. But if we do focus on those non-singleton fungal taxa, we find that they have a median of 6 or a mean of 18 plant species per fungus that they are able to colonize and on which we found. So each of these fungi is able to colonize. Some of them seem to be more limited to single species, but many of them are able to colonize some subset of the plant species available to them. If we look at the overall diversity of plants in these 3 habitats, we see a large number of species in the blue circles for the farm, somewhat smaller for the meadow, and even smaller for the forest. If we look at that same kind of Venn diagram for the fungi, it's not too terribly different, except the diversity of forest fungi is greater than that of the meadow, but the farm still provides the vast majority of the fungal diversity that we found. Taken together, we studied 476 different plant species and found 1,228 fungal OTUs distributed across them, with an average of between 2 and 6 times more fungi than plant species in a particular habitat. If we look at the intersection here for the plants, we see that about 15% of all the plant species were found in more than one habitat. Significantly more than that, 41% of all the fungi were found in 2 or 3 habitats. And if we focus in on that center part, what's sometimes called the Zeta 3 score, where these are the 2% of all the plant taxa that were found in all 3 habitats, and almost 20% of all the fungi were found in all 3 habitats. So what this is telling us is that the fungi are significantly less habitat specialized than are the plants on which they're growing. We're also interested in fungal diversity within the hosts. And so these histograms show on the x-axis the number of fungal OTUs, number of fungal taxa in, again, log2 scale bins, and the number of host plant species within each of those diversity bins. You can see they're all fairly similar across the different habitats, and that on the left-hand side, a very few host species are colonized by very few kinds of fungi. A few host species, plant species, are colonized by many, many kinds of fungi, but most of them are somewhere in the middle, between 1 and 3 dozen fungal taxa on a plant species. On average, it's around 2 dozen fungi per host species. Now, because fungi are able to colonize multiple host species, and host species are colonized by multiple fungal species, that creates a network structure. On the bottom of this network are the plant species in green. On the top are the fungal species in blue. And if a fungus is found in a plant, it is connected with one of these lines. And we see this tremendously complex network structure here. The fat blue lines are those fungi that are found on quite a number of different host species. They're much broader generalists, and they come from, these are the ascomycetes in the meadows, and they come from fungal genera that we would expect to see in these leaves and to be quite generalized. We can look at other guilds than these ascomycetes. In this case, these are the smut fungi and their yeast allies, including these 3 species of Malassezia that are found on a number of different host species, often 2 or 3 different Malassesias on the same host species. About 2/3 of the meadow plants are showing these yeast species. Now, this whole group of fungi is not very well studied, in part because they're very difficult to grow in culture. So they're only available to us through these kinds of metabarcoding sequences. And finally, if we look at this group of the rust fungi and their allies, these biotrophic, highly specialized fungi on plants, we see that they are connected to a fairly limited number of hosts, sometimes apparently rather specialized, sometimes with a handful of different host species. And this is what we'd expect with these biotrophic fungi. So we're seeing some really interesting patterns by using these metabarcode approaches. We're finding more fungi than plants overlap among the habitats, 2 to 6 times more fungal taxa than plant species in a community. And each plant species has about 2 dozen fungal associates with it. And most of those fungal taxa are found on some subset of the plant hosts in the community. This work was not possible without support from funding agencies and some amazing teams in the field led by Elizabeth Davis with a large number of students, and in the lab by Asa Conover and with his excellent team. So thank you very much.